So I wanted to continue on with the methods and sampling and analysis. Wanted to bring your attention to this idea where this is gaining traction in terms of microbial ecology, not just in macroecology. And so this idea that rare species in a mixed population could be contributing significantly to the ecosystem dynamics. And so keep this in mind. The big picture idea is that they're starting to look at this more carefully. They're starting to look at the activities of rare species, or maybe a better way to phrase that is less abundant species, number of species, individuals of that species. And so looking at their activities, their gene expression patterns, what are they doing? And they're starting to realize that these lower populations of individuals are actually contributing significantly, even in the microbial world. Here this is part of the section taken from that. And so, it's known that soil microorganisms are extremely diverse with relatively few abundant species and a large number of rare species. So there's a handful where there's lots of members of that species, but then there's many, many, many different species where it's very modest or low numbers. And they're starting to look at this more carefully. This was a greenhouse study in soil grown tomatoes, I think, but where is the word there? Few studies have investigated rare taxa and aquatic systems, found that rare rather than abundant played an important role in ecosystems functions such as nutrient cycling, especially after a disturbance, and that they can provide a seed bank, so to speak, which may become dominant after disturbance. So they're saying that it can be a good thing to have many of these lower populations of species so that if there is a disturbance, they have the ability to come back up and start reseeding, so to speak, through their reproduction. Where's the other word? Next one, seed bank, there's one other thing. Next, or no, that is the last. Oh, there it is. It's up higher. Sorry. Okay, so here they're saying these rare taxa can include more metabolically active microbes than abundant taxa by looking at their RNA to DNA ratios. So remember, when you're looking at RNA, you're looking at gene expression, what genes are turned on. And so looking at these ratios can help you decide how active those microbes are. If there's a lot of RNA, that means their gene expression is up and cooking. And they may be keystone species. So I thought that was interesting that they're using some of the macroecology concepts and bringing it down into the microbial world, so to speak. Okay, so keep that in mind as we move forward. This will come back again as we keep going with. I have a few last things to say. This not in this slide set because we're done with this slide set, but let me get into the third slide set on methods and analysis. Reshare. So now, again, going back to the ideas of macroecology, and if you've taken intro to ecology or if you're in BI 310, you might be thinking about species diversity and how to assess that in different ecosystems. And so when they get into the microbial realm, they are asking the same question, can we use what we know in macroecology and do that for assessing microbial diversity? And so it's a couple of terms that you need to think about. Species richness is the number of species per area. Okay, so you're just counting how many species are here when you look at richness. All species are given the same weighting and is very useful for comparing habitats. It's very useful for tracking things over time. So this data is important and can give a lot of good information, but it is limited because you're just counting the number of species. You're not looking at how many individuals of each species are there. You're just looking at how many species are there. So when you get into diversity, you want to look at not only how many different species are there, but how many of each species are there. So you're looking at they refer to it as evenness. So how evenly distributed are the numbers of each individuals of these species? How even is that? Is it even or is it way out of whack? So in macroecology, when you're learning to assess species diversity, you look at the Shannon-Weiner equation. And this is something that computers can easily spit out. You put in the data, you can then get the numbers spit out through this equation. H prime is the species diversity. And the P sub I is the relative abundance of species I in the area. So you're looking at counting up how many individuals are in each species. And then you're also looking at total number of species. So this is one way to do it. Okay. So what this does is it gives you values when you put in your numbers. If you end up with a species diversity, the H prime, if you solve for that with your data and you get 0, that's telling you you have one species present, which in real world ideas just doesn't happen. If you take a real world sample, you're not going to get 0, but that's the extreme. And then as you work your way above 0, species diversities will increase with the number of species in the community, but usually doesn't go above the number 5 in biological systems. So that's kind of your range to think about. And it's useful for taking random samples from a large community. So taking many, many, many samples is a good idea, but that also makes you think about how to take samples and how many samples do I need to take. We have to think about that as well, because when you set up an experiment, one of the things that you should be planning is not only your experimental design, but then you have to figure out how many samples are you going to be taking. And that is driven by what kind of statistics you want to run. And you want really powerful statistics. So then you have to think about that and go, okay, well, if I want to analyze this data using this statistical test, what do I have to do to meet the requirements of that test? So it's good data. So you really, before you even start your experiment, you have to think about stats and you have to design your method to make sure that when you go to analyze the data, you don't get this, uh-oh, you didn't take enough samples. That's like a blow to the heart, get that information. So think ahead, plan ahead, do the stats, thoughts, and then go back and finish doing your experimental design. So let's look at some data. Here we have 3 habitats, and on the left you see there's species A, B, C, D, E, and they all total 1,000 individuals. But I think if you look at each habitat, you can see the numbers of individuals of each species is varying between them, among them. So habitat 1, you have lots of members of species A, very few of anything else. So these are rare. B, C, D, and E are the rare taxa in habitat 1, and you have like a dominant species A. Habitat 2 is even Steven. Everything's this, 200 individuals. And then habitat 3, they mix it up a little bit more modest difference between the different species in terms of overall numbers. So species richness is the number of species.
So are we comfortable saying there's 5 species in each habitat, right? A, B, C, D, E, 5 species. Do you think the species diversity is going to come out the same amongst the 3 habitats? No, very different. And so if you run those calculations, you end up with H prime, you can see the values are quite different. So you can see where habitat 1 is closer to 0 because you know, 960 individuals compared to 10 of all the others. That's pretty extreme. You're going to get close to a 0 value. And then with habitat 2, because it's so even, you see that gives you the highest species diversity. You have 200 of each species. And then habitat 3 is in between those two. So this is just one example of how would this be used to look at different habitats and compare them. So it's important. I mean, yes, we certainly want to know how many different species are there, and that's important, but getting at a little finer detail of how many of members of each species are there can be super important. And again, going back to that rare taxa idea, you have to think about that because habitat 1, it might be tempting, like the old way decades ago to think about habitat 1 would be, well, species A is driving everything because there's so many of them. So they must be the ones really impacting the ecosystem, which is, you can understand that thought process, but now more careful studies are showing that rare taxa can actually be super important and that we need to be thoughtful about that as we look at these different habitats. And maybe that's something we really need to think about. Maybe we need to make sure that those habitats don't go extinct, or I mean, those species don't go extinct. So these types of ideas, they're starting to get a little bit more careful look at these rare species and not just zoom in on the super abundant ones. Now, the next question is, well, how do you sample if you're trying to get at species diversity? How many samples from that habitat should you take? And let's go through a story problem. Let's say we have a community and there are a bunch of smiley faces, which I'll show you on the next slide. And there's 7 different smiley face species. And then we say, well, we're going to take a cork borer and we're going to sample this habitat of smiley faces. And one of the devices, one of the cork borers is 5 times larger than the other one. And then we're going to go ahead and sample that environment. And so we want to know, you know, how many samples should you take? And again, this is really important. You need to think about this and running stats. This becomes pretty critical in your experimental design. So panels A and C on the left here are all the different, you see they're color coded by the different colors. Those are different species. So there's 7 smiley face species. And the smaller cork borer up top, the bigger one down bottom. And you can see they went ahead and sampled their sites. And so if you count up the smiley faces in each of the cork borers, the big one and the small one, you get this graph where how many times did you sample is on the x-axis and how many species did you capture are on the y. So we said there's 7 total. And so you can see with the bigger, the 5x cork borer, you end up with only having to sample 3 times to get all 7 species. So in 3 samples, you've done it, which is great. The fewer samples you take is generally a good thing in terms of minimal disturbance to the ecosystem for microbes. And it's less effort, less time, less labor. The small cork borer, even with what, 8 sampling, they only got to detect 5 species. They didn't even make it to 7. So they're missing. They didn't know that there's 2 other species there unaccounted for with that sampling. So that is not ideal. If your question is how many species are here, the smaller cork borer missed it. The bigger one got it. But That's not always a larger sampling tool like that doesn't always. It can be good for some things and not so good for others. Let's put it that way. For example, in microbial sampling, when you're looking at microbes, if you use a large cork bore, you can't tell if those species are interacting because that distance to them could be huge. Remember the scaling issue. We're 1,000,000 times bigger than microbes. And for a microbe, just what, 3 millimeter difference is like a 2 kilometer difference to us. So if we use a large sampling, if one of your questions happens to be, when I take this sample, my assumption is these microbes know each other, they hang out with each other, they're interacting with each other. But if you have a large sampling tool, you can't say that with any robustness. And so that's a question you really can't ask if you have a large sampling tool. And so they'll often say, oh, that's a little too coarse for looking at interactions or microbial ecology, because that's where, we're looking at interactions. And then, of course, your larger sampling methods do disturb the system more dramatically. And then it can be hard if you want to take repeated sampling to that site over time. You're disturbing it so much that that's definitely introducing artifacts into the system and not a good idea. So yes, larger tools might get you how many species are there more quickly and more accurately, but there's some downsides to it. And so whenever you design an experiment in microbial ecology, you do have to think about that. You have to think about how you're going to sample, what are you going to use to sample, pros and cons, what kinds of questions can we answer if we take the sampling in a certain way. So I would argue that when you do microbial ecology, it's a little bit trickier than if you're doing macroecology work in terms of just putting thought into how you do your experimental method. If you have a bigger tool, you really cannot sample the exact same area twice without, you have a lot of artifacts in the system because you've disturbed it so much. Does that make sense? Okay. Also, habitats from a microbial perspective can be very heterogeneous. And that is pretty much taken as a rule of thumb. A natural habitat, you're going to have a lot of patchiness and inconsistencies to the habitat. So like with soil, that's why we often use a cork borer, because it's usually a long tool that you can go deeper down into the soil. And of course, cork borers you can design to be really long. And so taking samples at different depths is often a good idea when you take soil samples. Even if you run these experiments and you go, oh yeah, there's 5 species in this sample, in this little ecosystem. Again, okay, that's good to know, but what are they doing? Are they active in the community? Are they dormant? Are they going long-term dormant, like forming spores, or are they just kind of quiet and not doing much? And so all of these questions, especially when you're looking at interactions amongst organisms, you want to know if they're active or if they're just sitting there like a lump. Okay. And so this idea of getting information on the species is great, but usually we don't stop there. We want to know how they're interacting with other organisms. And then we have to be careful about how, like with soil samples, how the question is, how deep should we go? How shallow should we go? How many samples to take? What's the size? So another idea that's been introduced in microbial ecology that has some success, it's been a long-term thing in medical microbiology, and that is Koch's postulates. So this is named after Dr. Koch, who came up with it. And he was a medical microbiologist that came up with a very methodical way to determine what people are sick from, what's causing the disease. And so with without him doing this, I mean, prior to Coke, it was not a very good thought process. So he decided to really approach it in a very methodical way. And so he came up with this pretty much a 4-stage process. He said, okay, if that person is sick with a disease and you isolate the microbe from that person, it should be the same microbe found in all the people sick with the same symptoms and the same disease. You should get the same microbe that you can isolate from the sick patient. You should be able to grow it up, purify it. And then they usually turn to an animal like a mouse or rat and said, let's see if we take that isolated microbe, if we inoculate it into a mouse, it should cause the same disease as what we saw in the patient. Then, after you inoculate the mouse comes down with the same disease, then you go and you sample the mouse and re-isolate that microbe. So this is something that is commonly done and has been done now for over a century, but this was, when it first came out, a pretty novel idea. But this really advanced the process of determining pathogens that cause human disease. It was a big deal when Dr. Koch proposed this. So this is great. It's been a boon for medical microbiology and human diseases and helping to advance our understanding and how to limit human disease. But the question was, microbial ecologists were curious, can you do that in more like, if you're taking samples out in nature and are asking the question, can this, what is this microbe doing? Can you somehow do a Koch's postulate process? And so they have been trying that for quite a while now. And this table are some examples. I think I just took a part of the table. There's more to it. But these are some examples of scientists that were kind of looking at this idea. And really, number 1 and 2 were the most successful of these 4. Number 1 and 2 were a little bit more decisive in using the Koch's postulate successfully. So the nitrogen fixation idea, number 1, they were looking at this bacteria called Rhizobium. That's the genus name. And so this is the type of bacteria that hangs out with plant roots and can fix nitrogen. And so they got it from the root. Does that say root? Root? Does that say roof? Doesn't that look like roof? I never noticed that before. Nodule on the roof of the legume. No, root. Whoops. So anyways, so what did they do? They took soil. So this was a contrived study where they're like, okay, let's see what's going on with this rhizobium. So they took soil that didn't have any rhizobia bacteria there. And what they did was they put it back into the soil along with some legume plants. And they were able to show that, yes, indeed, this microbe does fix nitrogen with this legume. They were able to re-isolate it, purify it. Yep. And so number 1, that one did successfully go through the Koch's postulates, and it was a nice study. Number 2 is looking at trichloroethene, TCE degradation, which is a known pollutant and is abundant in our environment. This one was groundwater beneath an Air Force base. No surprise there that there's contaminants there. And you all know that K.I. Sawyer used to be an Air Force base, right? Sawyer International. Kind of smile that they named it Sawyer International. Anyways, yeah, it was an Air Force base. So I'm sure there's a fair bit of pollutants in that area, in the water as well as the soil. Okay, so what was going on here? So they were looking at inoculating that habitat where the TCE is, and they were able to show that only after they put the dihalococcioides into that water area that you had activity going on and breakdown of TCE. So those ideas are, one was published way back in the 30s, and this one was early 2000s. Many other examples are out there, but basically looking at the this idea of, so here, first, let me just draw your attention to this idea where they went to the field site and they took their sample back to the lab and did a bunch of stuff and ended up isolating a microbe, okay? And then they wanted to say, can this microbe do a certain thing back in the actual habitat they isolated it from? So that's where this dashed line is trying to, this IVKP, that's Koch's postulates. So they're trying to show that if you get all the way down to an isolated microbe and you want to know what is it doing in that habitat, you could re-inoculate. And if you do a careful enough experimental method, you could ask the question, what is it doing? And is it truly doing that when you put it back into the field site? Because when you look at this flow chart, in the notes section, it describes all the different initials here. But basically, it's a lot of testing that they're doing. Molecular testing, biochemical testing, omics. They're looking at, you can look at proteomics and metabolomics and so on. So basically, you know, if you get these isolated and purified cultures and you say, oh, wow, you know, I sequenced this DNA and it has all these genes and I've done a bunch of proteomics or metabolomics on it. Well, that's great. And you're getting to know your microbe and that's really good. But you still can't say with certainty that just because it has all that means it's doing that back up in the actual field. So if that's your question, then you should think about maybe some kind of Koch's postulate idea to see if you can get data to support that. Oh yeah, it can break down TCE. In the lab, I can see that it can do that in the terms of it's got the gene necessary and all of that. But so, okay, is it doing it in the actual habitat? Habitats are messy and complex. It might just hang out and go dormant in that habitat. Don't know. You know, it's got a lot of competition and other microbes to deal with, as well as getting eaten by larger organisms. They're food, you know, microbes are food for other organisms. So don't know for sure. So basically this is a flow chart, just again, kind of getting at what do you do once you take a sample and, you know, you can do all this fancy stuff, which is great. But you have to be careful about saying, oh, I can tell that it breaks down benzene. It must be doing that in the field site. You can't link that idea with like certainty until you try to actually get at that in re-inoculating some soil from the field. So I had a undergrad several years ago take some field soil samples from, you know, along the lakeshore, the bike path. Do you know about the former Dow chemical company used to have facilities there? And I don't know if you ever noticed, but when you bike by there on certain days, the smell, there's a very odd smell. Some days it can be quite strong. And anyways, we were taking soil samples from there. This was back when Dr. Anderton was alive up in Eags, and he helped my undergrad pinpoint some areas in there that he could go sample for probably hotspots for contaminated soil. And so he did this kind of version where he isolated and purified bacteria and fungi. And then we had sample soils from that Dow chemical site, and he inoculated pure cultures in there. And then Dr. Putman helped him analyze, did the chemical break down? I helped him analyze, did the microbes survive that and come back out? Could we repurify it? Could we break down the chemicals or the microbe? Could the microbe break down the chemicals? So again, we were doing a Koch's postulate version to try to get at that idea, and really nice study. And unfortunately, we didn't keep following through on that all the way. It would have been good to ramp up that experiment, but anyways, it is a process that's done in a lot of different situations. I tend to focus on soil because that's my interest, but you can do that with water and sediments and even going down into the core of the Earth's crust. All right, so methods, whoops, lots of ideas on how to sample, lots of things to think about, including what kind of stats do I want to run on this, and that's going to guide how many samples to take. The last slide set, by the way, that's posted in Educat, it says labeled as definitions. That's just for your own interest. I'm not going to quiz you on any of that, but jargon, can be annoying. So I put that in there just in case you're like, what does that even mean? They define it in that slide set. There's lots of definitions. So if you're wanting to know, excuse me, it's available. Also, the other thing too, I wanted to remind you is that in the readings and video section of Educat, there's postings now there's a chapter on sampling, methods of sampling that's posted in there now. You'll see a lot of the images that I had in the PowerPoint are from that chapter. So, and then it's my book, so I have it certain highlights and things, so you'll see where I've highlighted. So that'll help too, but so make sure that you're paying attention to that and reading through that as you look at the PowerPoints and think about what we're learning. And then I also have a chapter reading on microbial populations and habitats, which we're going to start next. And then there's some other things that I posted in there. So make sure you go into the readings and video sections as you're studying for the exam, because that information is also important. Whoops, I need to reshare. Okay. So you're going to get the light version of population ecology and habitats, very much so. This could take years to go over. So I'm going to give you some big picture ideas. And one of the big picture ideas that's been persisting is this: everything is everywhere, but the environment selects, especially when you get thinking about microbes. This is a very famous thought. A lot of microbiologists embrace this idea. There's been some challenges to this a little bit here and there, but I mean, yeah, microbes are everywhere. It's really, I can see where this is a thought process that's pretty ingrained and it has a lot of validity. So when you think about this idea of microbial populations, it's this idea that they're going to be interacting if they're in close enough proximity. Certainly you can have all kinds of competition amongst microbes, which leads to succession of populations. But and that's typically what you're taught in an ecology class, but so much of the data currently with some of the hot research going on, we're now realizing, yes, certainly there's competition. That's a thing for real, but there's also a lot of cooperation going on amongst microbial populations, and that we have to look at competition, yes, but also cooperation ideas. And the more they look into it, the more they see it in terms of cooperation. So we have both going on here, but this is kind of the standard way that they think about different levels of populations, where you start with your individual, and then you have members of that species, let's say, and then you have a couple of species interacting and hanging out, and then it gets bigger and bigger and bigger until you start using the word ecosystem. All right. And so this idea that you have populations coexisting, very often you want to look at their dynamics. So often we'll turn to the logistic equation for population growth, which don't panic, I'm not going to ask you to do math. So, but this is what we have going for the logistic equation. dn dt is equal to b times n minus d times n, which you could simplify and just say it's equal to n b minus d. So that's just simplifying the equation. All right, this is just a simplification of the previous thing. So the B is the birth rate, D is the death rate, and then N is your population density. And then this net reproductive rate
So we're looking at how quickly are they reproducing?
And so it's really birth minus death is looking at your reproductive rate. And that's what they give as the R value. So if you do that, if you replace R for the B minus D value, okay, that's what they're doing here. They're taking that R and they're saying, well, we know that R, the net reproductive rate is equal to birth minus death. So they're using that in place of the B minus D value. And so that's how they get this equation here. Make sense? So they're just kind of substituting here. And then they said, let's put in the carrying capacity idea. So K is carrying capacity. And so if you put in this value, K minus N divided by K, so that's what they're doing here. So that's looking at the unused capacity of the environment. So basically, there's an upper limit to what that environment can handle in terms of how many people or how many microbes can fit in this habitat, right? There's only so much it can take before things go wonky. And so they needed to factor that into it too. So I'm just trying to show you a little bit of how they're thinking about this and how they came up with this equation to try to explain population growth in a certain habitat with certain limitations to that habitat, a carrying capacity. So you often see this when you look at data, population data, where they're looking at time on the x-axis and your population on the y-axis. And you often get this S or sigmoid shaped curve if you plot it out. So over time with reproduction going on, you'll have this increase in an S-shaped pattern like that, the sigmoid curve. And then your carrying capacity is the K value they're pointing to. So that straight line is your carrying capacity of that habitat. So once you get there, it's like, okay, that's it. We can't, no more. You're done. So I'm sure in like BI 210, you get into some of these ideas, at least a little bit. So hopefully this isn't brand new to you, but I think, you've lived out a normal life and you're scientists and you're, thinking about, documentaries talk about this. This is not a unusual concept. So when you start thinking about your net reproductive rate, the R value, your net reproductive rate and the carrying capacity and all of that, there is what they refer to as an RK gradient. And it's a continuum. So they're like, there's R, and then there's K, and then there's a gradient in between, a continuum between. So R is your net reproductive rate, per capita rate of increase of the population. K is your carrying capacity of that particular habitat. So let's imagine population density is really low. So lots of habitat resources are available, don't have many organisms, microbes there. Then the rate of population change is dominated by R. How quickly can they reproduce? Net reproductive rate. Because they have a lot of resources, they don't have to worry about a carrying capacity for a while. All right. So it's just kind of driven by what's the reproductive rate if you have really low populations. On the other hand, if you have lots of members of that population in a certain habitat, then carrying capacity starts to really pressure. I think if you just kind of mull this over a little bit, it starts to make sense that just intuitively with what you've seen so far in your life when you think about organisms and ecosystems. So yes, lots of organisms in a certain habitat, carrying capacity is definitely going to be influencing what's happening. What they're finding at the microbial level, similar to the macro level, is that organisms tend to optimize and take, they're either closer to an R strategist or a K strategist when it comes to how they live their life in that ecosystem or habitat.
So it's kind of an either or. Either they're like really good at reproducing quickly, or they're just really good at conserving resources, getting their reproduction done, but at a slower level. I kind of think about the, what is that story? The rabbit and the hare, and then they're racing, that kind of idea, where the rabbits are the R strategists, and then is it the rat? What is it? No, what is it? Turtle. Sorry, turtle and the hare, right? Sorry. The turtles are the K strategists and the rabbits are the R strategists. So here's some data kind of looking at the ideas between the two, the R strategists versus K strategists, and some of their thoughts on what would their characteristics be. So their growth phase, if you're an R strategist, that means if you have a short growth phase, it just means you can reproduce really quickly. You don't have a long drawn out process. K strategists, on the other hand, they can have longer ones, not always, but yeah. Rate of growth in uncrowded conditions, you can see R strategists are very high, whereas K strategists maintain a low rate of growth. Even if resources are high, they tend to just kind of plod along and have a more stately way of living that phase of their life. Relative food allocation during transition from uncrowded to crowded. So as they go from uncrowded to crowded, they are strategists. They tend to go, we're getting really crowded, eek, and they tend to shift into spore production. Not everyone, but that is a really good strategy if you're an R strategist, because you just don't do well at high population densities. It doesn't work well for you. K strategists, on the other hand, they continue to grow, slower growth rate, but they just grow and maintain themselves. And so the population density, you tend to, with an R strategist, they can get high populations, they can get die off once the carrying capacity gets there. But they have enough population going into that they can usually survive if they start crashing, if everything goes up to the carrying capacity and things start to crash. Whereas K strategists, they can still grow and hold their own and stay active at higher population dynamics. If you take an R strategist and you enrich them, so let's say you get a flush of nutrients into a habitat, then they can usually kick right back into a really fast growth rate. K strategists, that doesn't seem to really trigger them to go any faster. If they get an enrichment of like a big flush of nutrients in a system, they'll tend to, they'll grow, but they're not gonna go flying off the handle and grow crazy fast. Catastrophic death can happen with R strategists. K strategists, not so much. R strategists tend to migrate if possible. So when they feel, so to speak, that their habitat is reaching the carrying capacity, if they can migrate out of there, they will. Of course, that means they have to be modal in some way. So there's limitations to that. K strategists, it can vary. So when they started looking at this from a microbial perspective, I mean, this was already known at the macro level. There are strategists, K strategists, but they wanted to know, can you do this in microbial ecology? And for the most part, it is a effective way to think about populations. You can start to see that there are strategists and K strategists at the microbial level. So for example, the streptomyces bacteria that I work with, they tend to be thought of as K strategists, even though they form spores. But because they have a long growth phase, they grow slowly. Even in uncrowded conditions, they're not super fast. They just grow slowly. And they have a complicated life cycle, many parts to it. They don't just get bigger in size and go through binary fission where they divide into. That's not really how they do it. They have this big complicated life cycle where in the end they form some spores. But on the other hand, an R strategist like E. coli that can double its population in 20 minutes. I mean, that would definitely be an R strategist idea, even though it can't form spores. So just be aware, it's not, you don't have to form spores to be an R strategist. Okay, I think we're gonna stop there. We have to stop. It's 10:50 already. Okay. We'll continue on with this tomorrow.